Welcome back, everyone. Today we're doing a deep dive into the world of MALDI-TOF.
Specifically, the Bruker Microflex.
Right. It's for identifying those tiny but mighty microorganisms.
And we have a fellow medical microbiologist who wants to really get a handle on it.
They're looking for that step-by-step, almost recipe-style algorithm.
To use this technology in their lab, yeah.
So consider this your cheat sheet to understanding and using this honestly revolutionary technique in a clinical setting.
And it's amazing how practical these sources are. They lay everything out so clearly.
It's like step 1, step 2, step 3.
Almost like a high-tech cookbook for identifying bacteria.
Exactly. So for those who might be new to this, what even is MALDI-TOF? Why is it such a game changer in microbiology?
Okay, so MALDI-TOF stands for Matrix-Assisted Laser Desorption Ionization Time of Flight. It's a mouthful, I know.
It is a bit of a mouthful.
But basically, it's a way to identify microorganisms based on their unique protein profiles. It's kind of like a fingerprint.
Oh, interesting. Like a fingerprint, but for bacteria and yeasts.
Exactly. And this method, it's super fast, it's super accurate, which is, critical in the medical field.
Absolutely. Because time is of the essence when you're dealing with potentially serious infections.
No kidding. A quick diagnosis can make all the difference.
So speaking of the process, our listeners' sources, they lay out a really detailed workflow for the Microflex. Where do we even begin with this whole thing?
Well, before you even think about analyzing samples, you have to make sure the Microflex is up to snuff.
Working properly.
Right, exactly. That's where daily validation comes in.
Okay.
And the star of the show for this is what they call the Bacterial Test Standard, or BTS.
The BTS. The Microflex, work-wise, document mentions this a lot.
Right.
What exactly is it, and what does it tell us about the instrument's performance?
It's like a standardized sample made from E. coli extract, and it contains this specific set of proteins that the Microflex should be able to detect consistently. And so if the instrument can identify those proteins in the BTS accurately, you know it's calibrated right and ready to analyze patient samples. It's like your daily quality control check.
Ah, I see. So you run this test on the machine before you start your day to make sure it's giving you good results.
Exactly. You got to be sure you can trust what it's telling you.
Right. Makes sense. Now, the next step in the workflow, according to the sources, is creating this thing called an import sheet.
Yeah, the import sheet. It might seem like a small detail, but it's actually super important.
The Microflex work-wise document is very specific about the format of this.
Right.
Why is the format so important?
Well, Microflex, it relies on this database to compare protein profiles of unknown samples.
Okay.
And the import sheet, it basically tells the instrument how to organize and read the data. Any errors in that format, it can mess up the whole identification.
That makes sense. So one tiny mistake in the import sheet, and you could end up with a misidentified microorganism.
Oh, yeah, for sure. You're basically speaking the same language as the machine. You got to get the grammar right.
It highlights how precise you have to be with this technology. Every little detail matters.
Precisely. And that attention to detail, it carries over into the next stage, sample preparation.
Oh, right. Sample preparation.
There are actually 3 different methods outlined in the Microflex: work-wise document from back-check bottles, a direct method, and an extraction method.
3 different methods. Wow. Can you give us a quick overview of each one? When would you use which method?
Okay, so if you're working with, let's say, blood cultures, in those back-check bottles,
Right.
there's a specific procedure where you use a solution to break down the red blood cells and concentrate the bacteria. But, and this is important, the document really stresses this, you can't use the direct identification method on any potential class 3 pathogens from those bottles.
So safety first. Makes sense.
You'd have to work with cultures from plates in those cases.
Got it. What about the direct method? When would you use that?
The direct method, that's your most straightforward one. You use it when you have a nice isolated colony on a plate.
Okay.
You just take a tiny bit of that colony and smear it onto what they call the target plate.
So it's quick. Simple.
It is, but it doesn't work for every single microorganism.
And that's where the extraction method comes in.
Exactly. Some bacteria and yeasts, they need a little extra oomph to release their proteins.
Ah, right.
So the extraction method uses specific reagents, like formic acid, acetonitrile.
What do those do?
They break down those cell walls, make those proteins accessible for analysis, but got to work quickly during this step.
Oh, why is that?
Well, I'm sure you know this from your own lab work.
Oh, yeah. I'm guessing it's to prevent the proteins from, degrading or changing their structure.
Bingo.
Yeah.
You don't want those proteins to break down. You wouldn't get an accurate reading.
Right. It's like this delicate dance between speed and precision to get those samples just right.
Exactly. You've got to be quick, but you've also got to be careful. Those proteins are delicate little things.
So once you have those samples all prepped and ready, what happens next?
Then you load that target plate, the one with the samples, into the Microflex for measurement. And wouldn't you know it, there are separate SOPs for everything. Loading, unloading, even measuring.
Oh, wow. It seems like every little detail matters when you're working with this level of technology.
It really does. Every step has to be followed precisely.
What about cleaning the target plate? After it's been measured, that's got to be important too.
Oh, you bet. They outline a very specific cleaning process. Ethanol, trifluoroacetic acid.
TFA.
Yeah, TFA. They even warn you, don't leave the plate in alcohol for too long. Could damage the magnet underneath. It's amazing how something so seemingly small can have such a big impact.
It really does highlight how sensitive this technology is. So we've talked about daily validation, creating the import sheet, prepping those samples, even cleaning. Anything else before we move on?
Just a few end-of-day things, like a process they call specroname for converting the data, and a little reminder to lock that computer and restart it on Tuesdays. Even high-tech lab instruments need their weekly reboot.
It's true. Well, I think we've covered a lot already, making sure the Microflex is ready to go, how to format the data, how to prep the samples. It's amazing how much detail goes into each step.
It really highlights the level of, precision, the care needed when you're using MALDI-TOF to do microbial identification.
Absolutely.
But we're just getting started. Next time, we're going to tackle the preparation of all those reagents for both sample preparation and validation. Get ready for some very precise recipes.
That sounds fun. Can't wait. Welcome back to the deep dive. Last time, remember, we were talking about all those kind of intricate steps for getting the Microflex ready to analyze samples.
All those pre-game rituals.
Right, exactly. Now, we're going to be diving into the world of reagent preparation, guided by MALDI Biotyper Bilage 1, Breeding Reagentia, Forheen Microflex KML.
The recipe book.
Yeah. It's amazing these sources really provide detailed recipes for each reagent. It's like they know how crucial getting these right is for accurate results.
Oh, absolutely. Just like a chef needs the right ingredients for a successful dish, you know? Accurate and consistent reagent preparation is key for reliable MALDI-TOF analysis. One slight misstep could throw off the whole thing.
It's true. So let's start with the matrix solution. What's its role in all of this?
The matrix solution, think of it as the unsung hero of MALDI-TOF.
Okay.
It's a special solution, absorbs the laser energy during analysis, helps the protein molecules ionize,
Which is?
essential for them to be detected by the instrument. You know, the document specifies using these HCCA matrix crystals.
HCCA, yeah.
And they have to be completely dissolved in a specific solution. Acetonitrile, water, and trifluoroacetic acid.
Trifluoroacetic acid, yeah.
And they even emphasize, vortexing that solution for at least 2 minutes.
Oh, yeah. I've definitely been guilty of skimping on the vortex time in the past.
I get it, but in this case, those 2 minutes, they're crucial. Thorough mixing. It ensures those HCCA crystals are completely dissolved, evenly distributed. Otherwise, you risk having inconsistencies in your analysis.
Ah, so it's all about creating a homogeneous mixture for reliable results.
Exactly. You want that matrix solution to be perfect every single time.
Now, how long does this matrix solution last once you've prepared it?
Good news is it's pretty stable. The source says you can store it at room temperature for 2 weeks.
That is handy. Okay, next region on the list, the Bacterial Test Standard, or BTS.
Our old friend.
Yeah. As we talked about, it plays a crucial role in validating the Microflex. What makes its preparation so, I guess, special?
With the BTS, it's all about precision.
Yeah.
And protecting those precious proteins from damage, the document really stresses handling it carefully to prevent oxidation, which could totally affect its performance. No vigorous shaking or vortexing allowed.
Wow. It even says to avoid creating air bubbles during pipetting. They really aren't messing around with this gentle handling.
Nope. Remember, the BTS contains specific E. coli proteins. They're like the reference point for the Microflex. Any changes to those proteins can mess up the whole validation process.
So you prepare it, then you carefully aliquot it and store it at a negative 20 degrees Celsius.
Exactly. Got to keep it happy.
It's amazing how even something as, I guess, seemingly simple as pipetting can have an impact on the accuracy of the results.
Yeah.
Okay, let's move on to preparing formic acid, Fa. at 70%.
Formic acid, important stuff.
Yeah, the source mentions it's used in sample preparation, especially for yeast and gram-positive bacteria. Why those specifically?
Well, those types of microorganisms, they tend to have thicker cell walls, makes it harder to extract the proteins you need.
Oh, I see.
Formic acid helps break down those walls, releases those proteins more efficiently.
It's like giving them a little boost to make sure their protein fingerprint is clear for the Microflex to read.
Precisely. But of course, safety first. Always.
Absolutely.
Document reminds us to wear gloves, work in a fume hood when handling formic acid. And never forget that rule. Always add acid to water, not the other way around.
Yeah, safety first. Similar precautions apply when you're preparing Trifluoroacetic Acid, TFA, at 80%, right?
You got it. Both acids, you got to handle them with care and dilute them precisely for safety and to make sure you're prepping them correctly.
Okay. Last but not least, we've got Saponine at a 5% concentration. Now, the document says it's used specifically for preparing samples from BACTEC blood culture bottles. What's its role?
Saponine, it helps to last or break down the red blood cells in those bottles. This basically concentrates the bacteria, makes it easier for the Microflex to detect their protein profiles.
So it's like clearing away the clutter to get a better look at the bacteria that are in there.
Exactly. It's all about getting the best possible signal.
It's amazing how each region has such a specific role to play.
It really is. And thankfully, the source provides separate SOPs for preparing each region, nice and detailed, so you can follow along step by step.
Very helpful. It seems like precision and proper storage are like key themes in reagent preparation. Would you agree?
Oh, absolutely. From carefully measuring each component to storing those prepared reagents the right way, every step is meant to keep those reagents stable, make sure they work the way they're supposed to.
It's a constant reminder that with MALDI-TOF, the devil is in the details.
You said it. Every little thing matters.
Well, I think we've covered a lot today. From the matrix solution to the importance of saponine in blood cultures, it's clear that accurate and consistent reagent prep is like the foundation for reliable identification, using the Microflex.
It's the bedrock. You can't build a good house without a strong foundation.
Exactly. But our journey with the Microflex isn't over yet. Next time, we'll dive into the critical steps of validation and calibration, making sure your instrument is performing at its best. Giving you that confidence to trust those microbial IDs.
We're going to make sure your microbial detective is ready to solve those cases.
Can't wait. Welcome back to the Deep Dive. We've covered so much ground, haven't we?
Yeah, it's been a whirlwind tour.
We've talked about prepping the Microflex, whipped up some reagent recipes, but now it's time to make sure this instrument is really performing at its peak.
Make sure it's firing on all cylinders.
Exactly. We're diving into the validation and calibration process, guided by Microflex Validation 2, Validati en Calibrati von der Microflex.
Yeah, this is where we separate the wheat from the chaff, so to speak, the well-functioning instruments from those that need a little TLC.
It's like making sure our high-tech microbial detective is ready to solve the case.
Ready to get to work.
The document, it really emphasizes this distinction between validation and calibration. Can you break that down for us?
Right. Think of validation as your daily checkup. Just a routine thing to confirm that the Microflex is working right, producing good results.
Okay. Yeah.
Calibration, on the other hand, that's more like fine-tuning.
Ah, okay.
You're adjusting the instrument to make sure it's accurate, usually after maintenance or cleaning.
So validation is like checking your vitals. And calibration is like getting an adjustment, making sure everything's aligned.
Perfect analogy.
And for daily validation, our trusty Bacterial Test Standard, BTS, takes center stage again.
Can't get away from it.
Right. So the document, it says you apply the BTS to the target plate and analyze it, like giving the Microflex a known sample, making sure it can identify those E. coli proteins accurately.
Exactly. It's got to pass the test.
And it mentions marking the vial every time you thaw the BTS.
Yep,
Why is that so important?
Ah, that's a good catch. The BTS, it's got a limited number of those freeze-thaw cycles before its performance might start to, go downhill.
Okay.
Marking the vial helps you keep track of those cycles. Make sure you're using a fresh BTS.
Makes sense. Got to keep it fresh.
Yeah.
Now, what if the validation fails? Does that mean the Microflex is out of commission?
Not necessarily. First, try repeating it with a fresh spot of BTS. If it still fails, then it might be time for some troubleshooting. Could involve what they call a source cleaning. It's like a deep clean for the instrument.
Ah.
Or you might have to call in the experts, Bruker tech support.
So it's like a step-by-step process to try to isolate the problem. Now, you mentioned calibration. It's usually done after maintenance.
Yeah.
like a source cleaning. Does it also involve the BTS?
Oh, absolutely. The BTS is key for both automatic and manual calibration methods. Automatic calibration, that's your more straightforward one, relies on the Microflex's software to make adjustments based on the BTS analysis.
Okay.
Manual calibration, that's usually needed for more fine-tuned adjustments, especially after certain types of maintenance. It involves using specific software commands to tweak the instrument settings.
Wow. So precise. So both validation and calibration, they're crucial for keeping the Microflex in top shape.
They're essential. You want your microbial detector to have the best tools and training.
Right. Now let's put all this knowledge to the test with some real-world scenarios. We've got 5 hypothetical case examples to see how the Microflex would be used in a clinical setting.
Time for some action.
I love case examples. Okay,
Right.
Case 1, you have a positive Bactec blood culture bottle. Under the microscope, you see gram-positive cucky in clusters.
Classic Staphylococcus aureus.
Right. So you'd process the sample using Saponine to lely those red blood cells, concentrate the bacteria,
Get those bugs ready for their close-up.
and then analyze it on the Microflex, hoping for a nice, clear identification, high log score, to confirm Staphylococcus aureus.
That's the goal.
Okay, Case 2, positive Bactec bottle again. But this time, it looks like a mixed culture. More than one type of bacteria is present. What would you do?
Mixed cultures. Always a challenge.
Right.
Since the Microflex might not be able to ID everything in a mixed sample, I'd culture the bacteria on agar plates. Analyze individual colonies.
So, separating the suspects to get a clearer picture.
Divide and conquer.
Case 3, you've got a colony. From a urine culture, run it on the Microflex using the direct method. But the log score is low. Doesn't seem very reliable. What's your next move?
Low log score. Not ideal.
What would you do?
Well, remember how we talked about the extraction method?
Oh, right, to get a more concentrated protein sample.
Exactly. It might give you a clearer result.
Okay, good thinking. Case 4, you have a yeast isolate. What special considerations do we need to keep in mind for yeasts?
Yeasts, they're a bit finicky. They often need that formic acid during sample prep.
Right, to help release those proteins?
Yep. Gotta coax them out sometimes.
Last case, Case 5. You're all set to run some samples, but the validation process fails. What do you do?
Back to the drawing board.
What are the steps?
First, you repeat the validation. Fresh BTS if it fails again. Troubleshooting time, either that source cleaning or call Bruker.
You got it. Mastered the art of troubleshooting. These case studies really highlight how versatile the Microflex is,
It's a workhorse.
but also how important it is to follow those procedures and guidelines. It's like being a detective, isn't it? Piecing together clues, knowing how to handle any unexpected twists. But now, to really solidify our knowledge, how about some exam-style questions?
Oh, you're gonna quiz me.
I am. I've got 10 multiple-choice questions ready, covering all the key aspects of MALDI-TOF and Microflex operation that we've talked about.
All right, hit me with your best shot.
Listeners, are you ready? Let's see what you've learned. Question 1. What does MALDI-TOF stand for? A. Microbial analysis of lipids and DNA by time of flight. B. Matrix-assisted laser desorption ionization time of flight. C. Mass analysis of light diffraction and intensity by time of flight. Or D. Molecular assessment of living and dead isolates by time of flight.
That's a tricky one, but I'm gonna go with B, matrix-assisted laser desorption ionization time of flight.
You are absolutely correct. It's a mouthful, but it accurately describes the technology. Okay, question 2. Which of these is not a sample preparation method for the Microflex? A. Direct method. B. Extraction method.
Yeah.
C. Dilution method. Or D. From Bactec bottles.
I'm gonna say C, dilution method. I don't recall that one being mentioned.
You are on a roll. Dilution is not a specific method outlined in the source. Okay, question 3. What is the main purpose of the matrix solution in MALDI-TOF analysis? A. To preserve the protein samples.
Right.
B. To sterilize the target plate. C. To enhance laser absorption and protein ionization. Or D. To break down bacterial cell walls.
I remember this one. The matrix solution helps with laser absorption and protein ionization, so C.
Correct again. The matrix is essential for that initial step of getting those protein molecules ready for analysis. All right, question 4. Which reagent is specifically used for lysing red blood cells in Bactec blood culture bottles? A. Formic acid. B. Acetonitrile. C. Saponine. Or D. Trifluoroacetic acid.
That's Saponin, right? Option C.
They're a MALDI-TOF whiz. Saponine is our red blood cell buster. Okay, on to question 5. The Bacterial Test Standard, BTS, is primarily used for what purpose? A. Identifying unknown microorganisms. B. Calibrating the instrument after maintenance. C. Validating the instrument's performance daily. Or D, preparing the matrix solution?
The BTS is all about validation, so C.
You're on fire. Daily validation with the BTS is crucial for quality control. All right, let's make these last few rapid fire. Question 6. True or false? The direct method of sample preparation is suitable for all microorganisms.
False. Some microbes need that extra help from the extraction method.
You're right. Question 7. What should you do if the daily validation with the BTS fails on the first attempt?
Repeat the validation with a fresh spot of BTS, then troubleshoot further if necessary.
Excellent. Question 8. What is the maximum acceptable deviation in ppm during manual calibration of the Microflex?
300 ppm, if I recall correctly.
Right again. Question 9. Why is it important to handle the BTS carefully and avoid excessive freeze-thaw cycles?
To prevent protein degradation, which could affect its performance and the accuracy of the validation process.
Perfect. Last question, question 10. Why is it important to work quickly when performing the extraction method of sample preparation?
Speed is key to prevent protein degradation and ensure accurate results.
You aced the quiz. Amazing job. Give yourself a pat on the back. That was a whirlwind tour of the MALDI Biotyper. From understanding the basics to those intricate procedures, you've now got a solid grasp of this amazing technology.
We covered a lot of ground. Validation, sample prep, reagents, troubleshooting. It can feel overwhelming at first, but once you break it down step by step, it's totally manageable.
And remember, this deep dive, it's just the beginning. There's always more to learn, more to explore. So keep digging into those source documents. Experiment in the lab.
Practice makes perfect, right?
Exactly. And don't forget to share your insights, your questions in the comments below.
We love hearing from you.
And if you enjoyed this deep dive, be sure to give us a like. Subscribe to our channel for more insightful content. We're here to help you unlock the secrets of the scientific world.
Keep those brains buzzing.